Very great day, students. Already we discussed all about biomedical instrumentation subjects and all. Before it, you people learned the things and concepts of biomedical instrumentation. And here I want to recall all the concepts what we already studied before mid-exam. What are the electrical activities of excitable cells, functional organization of the peripheral nervous system, electrocardiogram, electroencephalogram, electromyogram, electroneurogram, and electrode-electrolyte interface polarization, polarizable and non-polarizable electrodes. In this, again, we have surface, practical hints of electrodes, skin electrodes, characteristics of bioamplifiers. All these are regarding the main concepts of biomedical instrumentation blocks, which are important for acquiring the different biological potentials. And what are the parameters and what are the ranges? If we want to diagnose about electrical activity of heart, the signal is pronounced as electrocardiogram, and the amplitude range is 1 millivolts to 5 millivolts, and the frequency range is 0.05 hertz to 120 hertz. And the electrical activity of the brain, EEG, electroencephalogram, its amplitude range is 2 microvolts to 200 microvolts, and it's typically 50 volts, and the frequency range is 0.5 hertz to 70 hertz. Next, coming to the nerve condition and muscle activities, EMG, electromyogram, is 25 microvolts to 5000 microvolts, and the frequency range is 5 hertz to 2000 hertz. Electrical activity of the eyes, it's known as electrooculogram. It's the potential due to movement of eyeballs actually, and the amplitude range is 10 microvolts to 3500 microvolts. The frequency range is DC to 100 hertz. The basics of biomedical instrumentation system. The patient, it is nothing but input stage, and the subject where we want to diagnose are doing experimental and all. So it's nothing but a primary sensing element. It's sensed by transducer or electrode, and this is amplified by instrumentation amplifiers. After that, signal processing unit, and this signal processing unit, we want anything into the digital form, right? So for that we will use here RTD converters, filters and all. And next, the system computer, maybe in output display, a printer, a surgical tool, or whatever it may be. These are the different blocks of biomedical instrumentation system. And next, excitable cells exist in nervous, muscular and granular tissue, exhibit a testing potential and an action potential necessary for information transfer. For example, sensory information in nervous system or coordination of blood pumping in the heart. And the different state of potentials are resting state as well as active state. Resting state study electrical potential of difference between internal and external environment, typically between -72 to -90 millivolts, relatively external medium. And active state is electrical response to adequate simulation, consists of all or none action potential after the cell threshold potential has been reached. By this pictorial representation, how action potential and resting potentials will occur. The ranges is -90 millivolts to +20 millivolts. See here, the polarized cell consists of all negative energy or all negative potentials inside the cell membrane, and outside is surrounded by positive potentials. It's all around -90 millivolts, -22. And depolarized is nothing but action potential. It's typically 20 millivolts. In this, all positive will throw inside a cell membrane and negative will move towards the outside of the cell membrane. So how it will conversion from resting to action? First, all the cells wants to be in rest position, that is called polarized state. From this polarized state, slowly some excitation will occur to the particular tissue or the particular cell. Then it will slowly transfers that resting state to the active state, that is called polarized depolarization. And after that depolarization completion, again it wants to repolarization. So here the cycle is polarized, depolarized and repolarization. This is the cycle will occur for each and every cell in our organism. Recording of action potentials. For this we use uh, electrodes. See here the electronic stimulator which is applied to the outer side of the skin layer. From that it will be acquired and amplified through the instrumentation amplifier. Cell potential is a function of membrane permeability and concentration gradient of various molecules, potassium, calcium, and sodium. Equilibrium potential is the membrane potential at which a given molecule has no net movement across the membrane. Nernst equation is in volts actually and at the 37 degrees centigrade. This is all about our resting membrane and resting potentials. Here the potassium are intra and extracellular concentrations are denoted by I is intracellular concentration and O is outer cellular concentrations. Equilibrium membrane resistance potential when net current through the membrane is 0. So here E is denoted with potential at 37 degrees centigrade. It's a Nernst equation. P is a permeability coefficient of the given ion. Factors including ion flow across the membrane, diffusion gradients, inwardly directed electric field, membrane structure, active transport of ions against electrochemical gradient. This is about our Nernst equation, and remaining we will be discussed in our next class. Thank you.